Okay, similar sort of vein as my talk last year with insects and fungi, this year I'm covering insects and viruses. Gee, I wonder what the inspiration was. Because it is a huge field, I'm only be covering a handful of examples across the range. But you'll see just why it's a large topic to cover. I could easily talk all week about this, but I'm not going to. You only got me for an hour. Very swiftly, the basic structure of a Hemorrhagic and virus. They are not living per se, but they are complicated chemistry that can hijack the mechanics of an existing cell to make millions of copies of itself and then explode out in the environment to infect more cells. The ocean water, for example, can have 9 million viral particles per milliliter. A quarter of all the living organisms in the sea are killed by viruses every day, but most of those organisms are single celled and replacing themselves just as quickly. The viral structure can have DNA or RNA at its core.
You just a side stop here.
Sorry.
Brilliant. Absolutely good.
Okay. It has the shell made out of capsid proteins which fit together like jigsaw pieces and then fold up into a remarkably intricate structure. And it may or may not have a layer of lipid molecules around the outside of that, usually scavenge whatever cells it's parasitised. And it has the spike proteins, which must have been in the news all the year, which attaches to the outside of another cell and then gets drawn in to hijack the machinery. The spike proteins could be analogous, say, to an email reporting from me being sent to the secretary trying to scan them out of eBay gift cards, which happened this morning. And the actual shape of the viruses, they can be icosahedral, as in the case of the common cold virus. They can be helical, in the case of the Mosaic virus, or they can be bizarre combinations of the above, in the case of the T4 bacteriophage that parasitizes coliform bacteria. Viruses are divided up into families depending on what shape they are, whether it's DNA or RNA, and what kind of DNA or RNA they have at their core, and whether or not they've got an envelope of lipid proteins and so forth. There are new viruses being discovered every day, often by advanced genetic techniques that are currently being applied all over the place. So it includes all kinds of new viruses that only affect insects, or that insects are being other animals. They all vary widely in what range of hosts they'll infect, how deadly they are to the hosts. For example, an example of an extremely lethal virus would be rabies. Rabies infects all warm-blooded animals, and it will also reproduce in insect cell cultures, they're fun bacteria, and that kills anybody who's asymptomatic. Without the vaccine, there's only been one person in history who hasn't died a horrible death that is non-violently insane as the virus has destroyed their brain. A lot of viruses are much more subtle than that. They'll quietly insert themselves into a cell and just re-emerge every couple of years, for example, cold sores or chicken pox, and with minor symptoms sometimes, or more serious ones. And some viruses actually insert themselves into the genome of the host organism and then be stuck with them for the next 100 million years. For example, the human genome, 8% of that is viruses, or former viruses that are now a permanent part of our genomes. Some of them are very important. There's one that's vital for brain development, and there's another one that's the only reason that the placenta stops the mother's immune system from destroying the fetus. Onto the viruses that infect insects. One quite common group of insect viruses are baculoviruses. When the caterpillar eats a virus as food, and the virus survives the upline midgut and gets into the animal, into the heat lymph of the caterpillar, it'll then start reproducing wildly until the caterpillar is basically turned into a bag of pus. hanging off the bushes like the world's worst Christmas decorations. And when it explodes, it's basically spraying the viral-laden pus all over the leaves underneath them. It becomes a bit of a question because you can see hundreds of these caterpillars scattered around the bushes even here in Perth. So how does the virus survive when there's only a few surviving caterpillars around and they're not spraying the virus all over the place as they die? It turns out that most of the time the mother moth is passing down a sublethal infection to her eggs. So they won't get particularly ill until something triggers a full epidemic across the countryside. One of those factors is spite. If the caterpillar gets infected by another kind of insect virus, the bacterial virus production goes through the roof, and this happens. This is from a hawk moth, the Cassava crop in South America. That's bacterial viruses all over the world, and we mentioned some of them in the later slides. But they don't just turn the caterpillars into bags of pus. Some of them are a bit more subtle than that. This particular virus wants the same thing and wants to produce as much virus as possible. And the way it does it, it makes sure that the caterpillar doesn't pupate. It just gets bigger and bigger, so when it does eventually die, there's a lot more caterpillar and a lot more virus to spray around the countryside. Or if you want even more subtle, there's the virus that infects the moth Heliophysia. That makes the female moths mate more than once and keep calling for more males after they've already mated. Don't feel too superior because increased libido and poor impulse control is also a symptom of syphilis. And of course, you've got the other viruses that infect the other terrestrial arthropods, such as the iridovirus. Iridovirus, once it's started to reproduce inside the woodlouse, forms crystalline masses that make the woodlouse appear iridescent, almost metallic blue. Only this month was confirmed that iridovirus is found in Australia. The paper came out in the last couple of weeks. But we've also got examples from amber of woodlice that are also metallic blue inside the amber. So that's probably iridovirus. So keep your eye on it, you might see one of these around the Perth if you're lucky. And of course, because the viruses can be hugely infectious, causing mass mortality, they've got to be very post-specific and only affect one particular species. There is real interest in using them as biological control agents. It's not necessarily the first option in doing that sort of thing, as was the case with the European spruce sawfly, which turned up in North America in 1922, and probably became a real problem for the forestry industry in the northeast of North America, to the point that they spent millions of dollars and at least hundreds of millions of parasitoids in an attempt to control it. And then the situation and the problem went away with one accidentally introduced virus. They're pretty sure that the virus in question came in with one of the parasitoids they were experimenting with, but once the virus got established in North America, there was no more major problem at all with the spruce sawfly. In its native range, it looks like birds are actually responsible for spreading the virus around. Most likely they're feeding on the sawfly grubs and then spraying the dipping virus-laden goo over other branches to infect other sawflies. Was that a question?
Oh, no, actually, but I just thought about our local sawfly burgers, for instance, the lemon-coloured ones, put their bottoms in the air and exude noxious alkaloids, and the birds avoid them because of that. What's wrong with this?
I don't know why the birds seem to be fine eating the spruce sawfly, but it seems to be going out of its way to look like a spruce needle, so maybe just not as poisonous one that's been eating eucalyptus leaves. So, yes, but after the nuclear polyhedrosis virus arrived in North America, the problem went away. And of course, ones that have been deliberately used as biological control agents include the Spiroptera frugiperta nuclear polyhedrosis and the related virus, the spruce sawfly virus. which has been approved for use in Australia now that a fall armyworm has arrived here. I mean, it's only been 12 months or a little over that it was first reported from the far north of Queensland. It's already in Perth, it's already in Tasmania. It's a massively invasive pest. So having the virus to use as a biopesticide will probably make a big difference. There's a few problems with it. You can only use it for the first couple of instars, the first and second molts of the cat never had much effect. But it does help that the caterpillars are cannibalistic, so if they're eating their infected brothers and sisters, they're going to get a dose of the virus anyway. One problem with that, of course, is that they only start cannibalising each other if they're running out of food already. Yeah, so that is going to be a major problem over the next few years, unless we come up with some useful control strategies.
And what does that destroy, that virus?
Mostly in Australia, corn.
Yeah.
Corn? Okay.
Another pest that was being controlled with a virus until recently is the coconut rhino beetle. For the last 50 years, there's been a nudivvirus that's been used to control the populations of the beetle across the Pacific. It's a serious pest because it destroys all kinds of palm trees, which include coconuts, raffia, date palms, and it attacks fruit trees and other fruits, tropical fruits such as pineapples and so on. The beetles completely wiped out the coconut populations on some islands. So it's reached Hawaii a few years ago. It turned up in Papua New Guinea last year. It hopped across the Torres Strait into Australia's going to march right down the coast of Queensland, destroying all the coconut plantations and native palms, native tropical fruits plantations. It's going to be a real problem if they don't find a new strain of the nudivvirus that actually infects the current outbreaks. Because we've been using the same virus for 50 years, so now let's push the beetle into those individuals that are immune to the virus. So we're going to find a new virus to do the same job. Hopefully they can do that soon before the immune rhino beetles actually arrive in Australia.
So Daniel, we need a Delta nudivvirus?
That would be one way to put it, yes.
Totally close to something that keeps palm trees,
But jumping from the viruses that actually infect insects, we can go on to the viruses that infect plants and that are spread by insects. It's another major problem with agricultural control of pest insects are the diseases that the insects themselves are spreading. Anybody who's worked in the ag department will know how serious those can be. I'm only going to use a couple of examples here. In this case, we've got the silverleaf whitefly, which turns out is at least 20 different species that can't be told apart unless you get right down to the microscopic and genetic level, which is its own problem. And they feed on at least 500 different plants. So there you have a complete morass of different viruses and different versions of the silverleaf whitefly that are picking up viruses from one plant, transferring to a completely new crop. And if you're trying to control them with, for example, another parasitoid or another virus, if you're not targeting the right cryptic species of whitefly, it's not going to have much effect. And if you're feeding on a completely different plant than either or woods, you're not going to control it at all. That's just some of the viruses that the silverleaf whitefly is known to pass on to the plants that it's feeding on. The Cassava Mosaic viruses and the Cassava brown streak virus down the bottom there are particularly serious because cassava is a staple crop across large parts of Africa and South America. So when those viruses are getting spread by the silverleaf whitefly, you can get mass starvation. Scott Bauer at the USDA Agricultural Research Service put a lot of these photos up for free use on Wikipedia, and that's been quite useful as far as putting this PowerPoint together. So they're very nice photos. But yes, so this is just one or other 20 pests that is becoming a much larger pest because they're just mixing and matching all the different viruses, all the different host plants, it's churning over and over, and you get outbreaks jumping from poinsettia crops to the tomato crops. And yes,
Sorry, why is it whitefly when it's actually a pump?
I think it's because they usually have white wings. But yeah, but they,
Anything that wings can be a fly, is that fair?
yes.
Cover my
Well, you do have the blood-feeding flies that don't have wings. They won't be covering them. They're still called flies, even though they don't have wings. So, but yes, it's not, of course, just the whitefly that do this. You also have the accidental plant hoppers can transfer viruses. You have trips that can transfer viruses. You have the aphids that can transfer viruses, such as the green peach aphid. The green peach aphid is the one species. You don't have to worry about 99 cryptic species that are identical to it, at least that we know about so far. It also has a very wide diet. It's not just peaches, as you can see from the short list there. And it is also a vector of at least 100 known plant viruses. So some of those viruses do have some quite interesting interactions between the plant and the aphid. For example, there's if you come to a Mosaic virus, if the plant is infected by that virus, its smell produces an odour that the aphids find irresistible. So the aphids, even in total darkness, will converge upon the infected plant, but it tastes repellent. So once the aphids had one taste of the virus-infected plant, they'll then leave again and spread the virus they just picked up to the next plant. But there's also one other interaction that we discovered between the aphid and two different viruses. Potato leaf roll virus, when it infects a potato plant, produces a protein called P0. P0 suppresses the plant's immune system, and the virus can take off, replicating its minions within the plant. So when one of the green peach aphids is feeding on the infected potato plant, the same protein P0 suppresses the aphid's immune system. And when the aphid's immune system is suppressed, an entirely different virus, the Mysis persicae denser virus, itself proliferates. But when the virus is out of control in the aphids, they get their wings earlier in the season and fly off to spread the viruses to other plants. That's one example of some of the interactions between in this case, two viruses taking advantage of aphids and potatoes. Then of course we get on to animal viruses. There are a lot of these. One way to divide them up is between those viruses that spread mechanically, that don't actually replicate inside the insect vector, and those that can replicate inside the insect vector, as well as in whatever host the virus is being passed on to. There's a term, archovirus, arthropod-borne virus, for those viruses that can actually replicate inside the insect vector. The best known insect vector for viruses is, of course, the mosquito, responsible for more deaths than any other animal in the history of the human race. But one good example of a mechanically spread virus is myxomatosis. Myxomatosis is native to one of the Americas, where it makes the swamp rabbit and a South American rabbit somewhat ill, but it is lethal to the European rabbit. When the myxomatosis was introduced to Australia and attempted to control the rabbits over here, it initially didn't get very far, but then we had a good mosquito season. And within months, it has spread from the ACT to Queensland. And shortly after that, however, it was just 2 years later, somebody in France had heard about how successful it was over here in Australia and released infected rabbits in northern France. And within 2 years of that, it was all over Western Europe. Looks like it was deliberately introduced into the UK and a year later deliberately introduced into Ireland and decimated the Aboriginal populations there. That was a problem. It's contributed to the near extinction of the Iberian lynx, which feeds largely on rabbits. It was going to be a disaster for the rabbit populations across Europe. That Winston Churchill made it illegal to spread myxomatosis in the UK. Over here, of course, it was just, we're happy to back the rabbits. Lots of motions from the UK where Beatrix Pottles, I think. Yes, you might also remember, if you've read it, in Watership Down, the white blindness that's described there is myxomatosis, because amongst the symptoms of myxomatosis is, of course, the horrendous eye damage and infections. So it's as a mechanically spread virus. It's not spread by the mosquitoes, so it's spread by fleas, mice, lice, and anything else that's feeding on the rabbits. Because it was a very good mosquito here, at the same time that myxomatosis was running rampant from the ACT up to Queensland, we also had an outbreak of the Murray River virus, which was making people very ill. So the researchers had to prove that it wasn't myxomatosis by injecting themselves with the myxoma virus.
Mechanical transmission, what do you mean? Is it just like the active feeding?
Yes, it is. Basically, they pick up the virus on their mouth parts. It's not replicating inside the insect. They're just passing it on as they feed, and the virus just has to stay active inside the insect long enough for that to happen. Now, when we go on to the arboviruses, the one that actually replicate, which I'm on to next, the situation of trying to control it becomes a lot more difficult because you have a permanent reserve of the virus replicating inside the insects as well as being passed on to the human or other hosts. But we'll get on to that now. Possibly the most significant of the arboviruses as far as the sheer amount of death that it's caused is yellow fever. It's one of the Flaviviridae. It's spread by Aedes aegypti and a few related species of mosquito. We're pretty sure that it originated somewhere in Central and Eastern Africa as a virus circulating between mosquitoes and monkeys. But once it jumps to the human populations, and it was from there spread across the world with the slave trade, it's become a global problem. Particularly in the Americas. Oddly enough, it hasn't barely got a foothold in Asia, possibly because Asia is already full with related viruses. So they've got the cross. If you get infected with one of those, you don't get yellow fever, which is not particularly ill. But yes, in the Americas, it has become called the American plague. Is that a question, sorry? Okay, fine, sorry. And it was getting to the Americas and elsewhere in the world as the mosquitoes were reproducing inside the fresh water supplies on board the ships. You'd have a generation of the mosquitoes hatch from the water supply, bite somebody quite often in the slave holes, infect them, then the next generation of mosquitoes pick up the virus and really be infected. This goes around and around. Some outbreaks are happening as far away as Glasgow, and it was spread in ships' water. Of course, these days, infected mosquitoes can also be spread on planes, which is a lot faster. Yes, the American plague. It was, until the invention of the vaccine, it was spectacularly lethal. The symptoms range from fever up to delirium and death in 60% of cases. Even the Mayans in that named the disease after one of the symptoms, the black bloody vomit. It's not an appalling disease. Some of the more memorable outbreaks in Philadelphia in 1793, 10% of the city were killed by yellow fever. They blamed that outbreak on a cargo of rotten bananas. The mosquitoes may well have come on the same ship, so not entirely off the right track. The first attempt to dig the Panama Canal, there were 20,000 deaths from yellow fever. Trying to avoid it was abandoned. They lost over half their top engineers on the project. The head engineer of the whole thing came to Panama with his family, built a luxurious house lost all his family members to yellow fever, shot his horses and went back to France. And during the Spanish-American War, they were losing 13 times as many people to yellow fever as they were to the actual combat. So there's a cartoon from 1905, Harper's Weekly, where the second attempt to make the Panama Canal was obvious to everybody that it was going to work at all. They needed to control the virus. Of course, they didn't know it was a virus at the time, which is how we get on to the next slide, which is some of the ways they tended to control yellow fever over the centuries. Initially, he had quarantine and cordon sanitaire. The yellow fever was one of the diseases that needed to be reported before your ship would be allowed to come to dock. The fact that the quarantine flag is yellow is probably a coincidence, but Yellow Jack, which was also the name on the cartoon there, was also the name of the yellow quarantine flag, so people did see a connection there, even if there wasn't one. They initially thought that yellow fever was spread directly person to person, but relatively early on they figured out that it couldn't be spread directly from person to person, because amongst other things, people trying to commit suicide after their family had died of the black vomit and so forth, by rolling in the black vomit and they hoped they'd get to die themselves. It didn't work, because of course it's being spread by the mosquitoes. Dr. Carlos Finlay in Cuba in 1881 was the first person to actually experiment with that hypothesis. He was doing some pretty good work, but it wasn't until Major Walter Reed, who did work in the US and in Cuba, actually proved that it was mosquitoes spreading it. Not least by noting that along the Potomac River, it was the troops who were actually walking home at night through the swamps that were getting yellow fever, and not the people that were drinking the swamp water or living in the nice mosquito-screening houses and so forth that were getting it. And when he came to Cuba, as part of the attempts to control it there, he actually got volunteers to get them to be bitten by infected mosquitoes to see if the mosquitoes are actually passing on the virus. This is also the first example of informed medical consent in medical experimentation, because he told these people, you could get ill, you could die in this experiment, and they volunteered anyway. It was also proved that it was actually a virus and not a bacteria, even though we've never even seen viruses at this point. Most viruses are so small, you can only see them with an electron microscope. Some are large enough that they originally thought to be very small bacteria, but most of them are so small that they go right through a sieve that sieves out anything even as small as a bacteria. That's one of the reasons that the original name for a virus was filterable virus, and there were some theories that a virus is actually a liquid life form. They're not, they're just very, very small. But it was Walter Reed proved that it was actually a virus that was causing the infection, and that it was spread by mosquitoes. The virus itself wasn't isolated until 1927, decades later. Major General William Gorgas was in charge of a major program trying to eliminate yellow fever from Cuba. His initial effort seemed based on the idea that the virus was spread by miasma, bad smells, infested houses and whatnot. So his initial attempt was blowing up supposedly infected houses and that, and burning them down, trying to clean all the garbage out of the streets of Havana. None of it worked, because he did nothing to stop the population of infected mosquitoes just breeding in the canals, ponds, plant pots, and whatnot. But once Walter Reed had published his results, says, we can actually control yellow fever now by going after mosquitoes. And he did. He had an entire corps of workers out there spraying oil on top of ditches and so forth, removing stagnant water from around Havana, and got yellow fever under control. And then you apply the same program to Panama, which is the only region that can lose another 20,000 people to yellow fever, finishing the canal. And I'm just in typo.
Disinfection.
Okay, yes. But the first vaccine was developed in the 1930s and earned a Nobel Prize for the work. It was actually suspected that the virus may pass down from mother mosquitoes to their eggs as early as the 30s, because mosquitoes had never been exposed to a human, never had an opportunity to bite and infect a human, but still capable of passing on the virus. But that wasn't proven until 40 years later in the 1970s. As I mentioned, because aircraft are a much faster way for mosquitoes to get around the world these days, aircraft disinfection has happened from the 1930s onwards. That photograph there is from a New Zealand airline. But yes, whenever, even these days, when you've got a plane coming through an area where yellow fever is known to be endemic, or Zika virus outbreaks and so forth, and other insect spread, the other viruses, they will fumigate the plane and the passengers.
They still do that now, don't they?
Yeah.
So I actually like disinfection quite well.
Yeah. So look, I like the way myself. Yeah.
I don't like to bother though.
Are you doing the talk next month?
No. Just the critique.
An example of a local pest, also a vector of other viruses. You've got the common banded mosquito, which is brownish, has a pale band around the process. It's found across most of Australia and quite a neighbouring countries and so forth. It is a vector of grave encephalitis, Fujian virus, Japanese encephalitis, also found in Australia. Ross River virus, Barber forest virus, as well as dog heartworm and the roundworm, Wuchereria bancrofti, is a major serious vector in all the parts of the world that it's found. It's just lucky that most of the viruses are spreading in Australia aren't nearly as lethal as, for example, yellow fever. It was first described from the Blue Mountains near Sydney, funnily enough, despite the fact that it's found in such a wide part of the world. One of the reasons that it is so widespread is that it'll breed in brackish water, it'll breed in purely fresh water, it doesn't even need a large amount of water breeding, just a cupful is more than enough for a tree hollow filled with rainwater. So it's a very widespread mosquito, it breeds anywhere, and it spreads a whole range of quite nasty viruses, including, of course, Ross River virus, the ones you get around Perth on a regular basis, such as down around the Peel Inlet. The Hemorrhagic encephalitis is the one that was spreading at the same time that myxomatosis was, and the researchers had to it's not myxomatosis that makes you sick, it's an entirely different virus, and they had to prove it on themselves. Of all the Flaviviridae, and there are a lot of them, there's only 3 of them that actually reproduce enough in a human host to infect another mosquito. Some of the other Flaviviridae will make you very sick, but they're not going to make the next mosquito that bites you sick. Danky Fever is an interesting one. It's very unpleasant. It's also known as breakbone fever from a joint and muscle pain that it causes, but you're probably not going to die from it. However, there are 4 different strains of dengue going around, and if you get a second different strain, you can quite likely die from your own immune reaction. So, but there are also a bunch of other viruses that only infect the insects and don't get passed on to human or other hosts. And some of them are very interesting, such as the 3 I've mentioned there, the self-fusing agent virus, the Hemorrhagic virus, and the Palm Creek virus. The last 2 were discovered in Australia because when a mosquito is infected with those viruses, it can't be infected with, for example, Ross River fever. It's immune to the Ross River virus if it's already got those insect viruses. So if they are also being researched as a possible control agent for Ross River virus, if you can deliberately infect the mosquito population with those viruses, you can stomp out the Ross River virus outbreaks. And you've got the other viruses in general arthropod-borne. You've got a subclass of that. You've got the tick-borne viruses, tick viruses. I'll only use one example here, but there's a lot of them as well. the Crimean Congo Hemorrhagic Fever. That's a fun one. Amongst the symptoms are massive bleeding underneath the skin, and liver failure, kidney failure, and death in up to 70% of cases. It's spread by not just one species of tick, but dozens of species of tick. Most of the virus replication tends to take place in the small mammalian hosts. Birds are mostly immune to the virus with the strange shift of ostriches. You can get very large amounts of the virus inside hooved livestock like goats and cattle and whatnot, but doesn't make them ill. But in humans, you can get from the ticks or from when they're butchering the livestock and that, it can kill up to 70% of the people who contract it. So, they originally thought there were different hemorrhagic fevers, but it turns out that the same virus that was turning up in the Crimea is the same as turning in the Congo as well. It's actually spread over a wide part of the world, possibly because the ticks are being carried around the world on birds. Okay, so they covered the viruses that infect insects. We covered the viruses that are spread around plants by insects. We covered the viruses that infect people and other large animals. Here we have some of the viruses that are used by insects. The polydNA viruses, ecolovirus and brachyvirus, which are spread by ignoring wasps and recruited wasps, are used to disable the immune systems of the caterpillars that they're parasitizing. The viruses are totally incorporated into the wasp's genome now, but the virus particles are only replicating inside the female wasp's ovaries. So when she's injecting her egg into the caterpillar, she's also infecting the polydNA virus, which shut down the caterpillar's immune response long enough for the egg to hatch and actually get going as a larva. There's a really neat set of experiments done with another one of the viruses, because of course the caterpillars aren't completely defenseless. Some of them are using a virus to kill the recruited wasps they're trying to parasitize them. The experiments were done by deliberately infecting the caterpillars with two bacillus viruses that were known to kill the recruited larvae. And yes, it certainly worked. If the caterpillar had these viruses in its hemolymph, then the parasites couldn't get started. But the same genes that are in this virus, in some species of caterpillar, are incorporated in the genome, so the caterpillar can make its own version of the parasitoid killing factor using what used to be a virus and is now not part of its own DNA. And that's the case with the beast armyworms, because they've already got the viral genes in their DNA. They don't need to be infected with the bacillus virus to protect themselves. And this works against most of the recruited parasites, but it doesn't work against the basket-occurring parasitoids, so Meteorus pulchricornis, which that's immune to the virus that kills the other recruited wasps. But the basket-occurring parasitoid injects the same virus into the caterpillars that kills other recruiters. That way, it's killing rival species of parasitoids by deliberately inoculating its own caterpillar host with a virus that kills other parasites. And you probably remember the fly destroyer fungus that I mentioned, that I covered in the insects and viruses talk last year. Well, it turns out it might be using a virus to control the fly's brain. They've discovered a strain of the fly destroyer fungus actually infects fruit flies, which is good for our research, and then discovered that the strain of the fungus is also carrying a virus from a family of viruses that are known to paralyze insect hosts, which is one of the things that the flies that are infected with the fly destroyer fungus are known to do. They'll find a suitable spot underneath the branch or on a window and just sit there paralysed until the virus has started to sprout. And the typhoid virus is known to be in the same family of viruses that do the same sort of thing to other species of insect. So for example, The Dinocampus coccinellinae paralysis virus is in the same family. It's used by the lady killer wasp. The parasite in question doesn't kill the ladybird when it emerges to pupate, but if it's just sitting there and it's a cocoon, it's vomiting other predators to come along. So what happens is that the ladybird that has been parasiting just stands guard over the cocoon. And if anything comes to try and harass the cocoon, the ladybird goes berserk, flailing at it with its legs, but never actually wandering off to go eat more aphids by itself. And we know that there's a virus doing this because it's an infravirus, and the virus is only replicating inside the brain, doing selective brain damage to the ladybird. But the interesting thing is that for the usual hosts for the lady killer wasp, about a quarter of the time after the wasp finished pupating and flown off, the lady beagle will get over the infection and go along about its business. It will recover from the viral infection of its brain.
That's like a timing thing as well. That's,
It's the same virus, it's the same wasp is on the cover of this National Geographic too. So, but that's only with the usual ladybird host for what this wasp. The other species that the same wasp can infect generally die. So obviously it's a bit of an arms race going between the wasp, the virus, and its usual host, and the other lady beetle is just a casualty. And that would be the end of the talk there. So I thought I'd had some up to later. Okay. I hope you found that generally informative and interesting. So as you can see, it's a non-stop arms race, a continually churning warfare for the last 300 million years. A virus is being used, abused, and incorporated. So it's I hope you found that interesting. Question, sorry.
Daniel, since we've all become epidemiologists, you didn't mention aerosol spread, really.
Okay, yes, I should have mentioned that. Because most of the insects aren't going around wheezing and sneezing and running a marathon and sucking down particles, most insects pick up the viruses they're infected with through feeding. And because the insect's ghastly outlines are as acidic as is with vertebrates, they need entirely different defences against the outline environment before they can get across the midgut lining into the body of the whatever insect is infected. And in some cases, then back into the salivary glands to be injected into the next plant or animal host for the virus. And because the insects don't have moist mucous membranes on the outside, it's only when they're being attacked by parasitoids or feeding on virus smeared plant matter or whatnot, they're picking up the virus or sucking up the virus along with infected blood.
Surely spherical is, it's a giant opening in the side compared to the side.
Very little actual volume of airflow through the holes. They're much lower metabolism as far as the respiration goes. They don't need to breathe as much. So, most insect viruses are sort of the midgut. So. Or injected along with the wasp egg. Any other questions?
do you think the Chinese will invent one of these?
Well, given that the rabies virus can replicate inside insect cell cultures, it probably wouldn't take much for a rabdovirus to have an insect vector if you've got, say, a few mutations that sort of actually can replicate inside an insect host as well as in a mammalian host as well. So yes, it could be. I could easily see at some point a baculovirus with an insect vector, which would be a nightmare, given that maybe it's just one of the baculoviruses and there's some really nasty viruses in that family. So.
Just on that, there was a Children Four Quarters program on Monday about the weaponised artificial intelligence, and one would think that this would be a complementary sort of development along with the horrors of little tiny drones which shoot fatal thoughts and that sort of thing. So it would take a particularly nasty government to do that, but it's conceivable.
Biowarfare research is and has been a thing for a long time, unfortunately. And indeed, some of the other, there wasn't a virus. Of course, during the Second World War, there was a Japanese research program for organising typhus and experimenting on human victims in China. So, yeah, you certainly use insect vectors of deadly diseases as a war weapon. So, it's been done. Yeah.
You're aware of maybe any viruses that affects the Placenta
Okay, most of the research has been done on the vectors of human disease or major crop pests, and a lot of the research is on caterpillars, possibly because it's not easy to mash up a caterpillar in a test tube than mash up a test tube full of aphids. But I can't think of any viruses that specifically infect Neisseria proprietor, but if Neisseria is vulnerable to the fly destroyer fungus, which is quite a wide host range, it's possible that the typhoid virus also infects, or some version of the typhoid virus also infects the blowflies to paralyse them for the virus, the fungus emerges. So, but yeah, every week there's new viruses being discovered, learning through genetic techniques. So, anything else? Anybody else? No? Okay then. Well, I hope you all found that enjoyable.
Daniel, what better way to spend his birthday than
If anybody has anything for show and tell, now's your chance. I may need to get going at this point because it's my birthday and you're not to be home. But yeah, I'll see you all at the next meeting. Our guest speaker next meeting is going to be Gavin Pitts. He's going to be covering his favourite parasites. So expect more body horror. Possibly along the same line you saw here. I'll try to make sure he doesn't double up.
We'll let you know on the very birthday.
Don't forget the knife.